In this lesson, we are exploring the different food tests that are required for your General Certificate of Secondary Education Biology Practicals. These include the Benedict's test for detecting sugars, the iodine test for starch, the Biuret test for proteins, and the Sudan-3 test for lipids. For those studying the Edexcel curriculum, we will also include the emulsion test, which is an alternative method for identifying lipids. All of these tests have a common starting point, which involves preparing a food sample. To do this, you begin by taking a small piece of the food item and breaking it down using a mortar and pestle to create a fine paste. You then place this crushed food into a beaker and add distilled water, stirring the mixture gently using a clean glass rod to help some of the contents dissolve. The next step is to filter the solution using a funnel lined with filter paper in order to remove any remaining solid particles. What you are left with is a clear liquid sample, which will be used for each of the tests. To test for reducing sugars, you use the Benedict's test. Reducing sugars are simple sugars that are able to react with Benedict's solution, though you do not need to memorize the distinction between reducing and non-reducing sugars at this level. To perform the test, take 5 cubic centimeters of your prepared food sample and place it in a test tube. Meanwhile, set up a water bath and heat it to a temperature of 75 degrees Celsius. Once the bath is ready, add approximately 10 drops of Benedict's solution to the test tube, then carefully place the test tube in the water bath for about 5 minutes. For safety reasons, always use a test tube holder and make sure the tube is pointed away from you and others to avoid accidents. If no reducing sugar is present, the solution will remain blue. However, if reducing sugars are present, the color will gradually change to green, yellow, or brick red, depending on how much sugar is in the sample. A stronger concentration of sugar results in a deeper red color. Next, we test for starch using the iodine test, which is much simpler to conduct. You start with 5 cubic centimeters of your prepared food sample, then add a few drops of iodine solution. The solution will initially appear as a browny orange color. After giving the test tube a gentle shake, if starch is present in the sample, the color will change from browny orange to a deep blue-black. Since this test only requires the addition of iodine, it is commonly known as the iodine test. The Biuret test is used to check for the presence of proteins. In this case, you use 2 cubic centimeters of your food sample and place it into a test tube, then add 2 cubic centimeters of Biuret solution. The mixture will begin as a blue color. After giving it a gentle shake, if proteins are present, the solution will change from blue to either pink or purple. If you are following the Edexcel exam board's instructions, you may need to be more specific by adding potassium hydroxide first, followed by copper sulfate, which together form the Biuret reagent that causes the color change when protein is detected. Now we move on to lipids, also known as fats and oils. For this, we use the Sudan-3 test. Take 5 cubic centimeters of your unfiltered food sample and place it in a test tube. It is important that the sample is unfiltered because lipids may be present in the solid particles. Add 3 drops of Sudan-3 stain solution and gently shake the mixture. If lipids are present, they will bind with the stain and separate from the rest of the mixture because lipids are not soluble in water. This will result in the formation of a bright red layer at the top of the liquid, clearly indicating the presence of lipids. For those studying the Edexcel course, you may be required to use the emulsion test as an alternative way to detect lipids. In this test, you take your food sample and add 2 cubic centimeters of ethanol, then shake it vigorously to allow any lipids to dissolve into the alcohol. After this, add an equal volume of distilled water to the mixture. If lipids are present, the solution will turn a cloudy white color, indicating that the lipids have formed an emulsion. If there is no lipid in the sample, the solution will remain clear. This concludes the food tests required for your biology practicals, and understanding each one helps you identify the presence of important nutrients in different food samples using simple chemical reactions.